I'd like to begin by thanking Professor Brunori for this kind introduction and to President Parisi for inviting me to give this lecture. This is a special pleasure for me to be able to talk about Linus Pauling in front of such a distinguished audience of intellectuals. It's also a pleasure for me because Pauling motivated an enormous amount of research in science and medicine in the 20th century, and also motivated much of my research for now over 50 years. Pauling is the only person to ever win two unshared Nobel Prizes, the Nobel Prize in Chemistry in 1954 and the Nobel Peace Prize in 1962. And as Professor Brunori pointed out, it's almost exactly 70 years since his famous science paper, which gave birth to what we now call molecular medicine. It was titled Sickle Cell Anemia, a Molecular Disease. So I thought I'd begin, because I suspect there may be some historians in the audience or physicians in the audience, to give what I call an ultra-brief history of human disease, the causes of human disease, what physicians call the etiology. So prior to Hippocrates, now this is ancient Egypt, disease was thought to be called by either evil spirits or it was a punishment by the gods. Hippocrates was the first person to reject this idea. He was known as the father of medicine. His idea was that disease was a natural phenomenon and it was caused by an imbalance of four humors: blood, yellow bile, black bile, and phlegm. This idea was promoted extensively by the famous physician Galen in the 2nd century, in the beginning of the 3rd century. And Galen thought that of the four humors, blood was the most dominant one, especially if one had a fever, and hence bloodletting with leeches. So that was the origin, actually, of bloodletting as a treatment for disease. Now, he was so influential because he wrote extensively. And his idea of, his concept of the four humors lasted for over 1,000 years. In fact, it was taught at the first medical school, the Scuola Medica Salernitana, if I pronounce it properly, which was founded in about the 11th century. And it was also because of this promotion by Galen, Salerno was known as the town of Hippocrates. The second medical school in the Western world was actually in Bologna, which was founded in 1219. Now, my favorite character in this history is Paracelsus. He called himself Paracelsus because I guess he got tired of telling people what his real name was. It was Philippus Aurelius Theophrastus Bombastus von Hohenheim. Now that was just too much of a mouthful, and he named himself Paracelsus, which means better than Celsus. Celsus was also a famous Roman physician. Paracelsus actually introduced chemistry to medicine, and that's how I got interested in him when I first read my first history of chemistry. He pointed out that the imbalance was not of humors, but there were chemical imbalances, especially minerals. And if there was too much of a particular chemical, that would become a poison. He also, however, believed in gods and astrology. And one of the things I learned today in the exhibit on Da Vinci was Da Vinci did not believe in astrology, but Paracelsus did. Not very much happened for several centuries until independently Louis Pasteur in France and Robert Koch in Germany showed that bacteria and other microorganisms could cause disease. And then in the early 1900s, a large group of scientists showed that viruses could cause disease. So here we are from evil spirits to viruses. So it's kind of interesting to think back at this because abnormal molecules were not considered until the middle of the 20th century with Pauline's paper on sickle cell disease. So first, let me tell you, it's often called sickle cell disease or sickle cell anemia. So let me just tell you briefly, what is sickle cell anemia? The protein hemoglobin, which is colored, it gives the color to red cells, is the protein that carries oxygen from the lungs to the tissues and carbon dioxide back to the lungs from the tissues. Normal red cells have a biconcave disc shape. They're red in the arteries where the hemoglobin is fully oxygenated, and when the oxygen is dissociated or partially dissociated, in the veins, they're more purplish color. In sickle cell disease, the red cells in the arteries are mostly biconcave disc shaped. They look perfectly normal. However, when the red cell becomes deoxygenated, they take on a variety of shapes, some of them having the shape of a sickle, and that's how the disease got its name. The disease is actually caused by these distorted red cells because they're much less flexible. And when they pass through the narrowest vessels of the tissues, called a capillary, they can become stuck to produce a logjam effect where no more cells can pass through the tissues, and therefore no oxygen can be delivered to this tissue surrounding the capillary. This has very severe consequences for patients. It causes acute and chronic organ damage, a shortened lifespan, and episodes of pain that are so severe that they're called a sickle cell crisis. Think of a kind of a myocardial infarction, but it occurs in every organ of the body. There are only 100,000 sickle cell patients in the United States, so it's called an orphan disease, but there are several million people worldwide who suffer from the disease, most in sub-Saharan Africa and also in India. And because they're third world countries, we really don't have very good epidemiological statistics. There's at least 3 million, and some estimates are up to 7, 8, 9, 10 million. In parts to try to show you how severe the disease is, in parts of Nigeria, 95% of children born with the disease die before age 5. So this is the legend of how Pauline learned about sickle cell disease. In 1945, Franklin D. Roosevelt appointed a committee of physicians plus one scientist, Linus Pauling, to advise the federal government on how they could assist in scientific and medical research following the Second World War. And this was the foundation of the National Institutes of Health and the National Science Foundation. Castle was a famous hematologist at Harvard Medical School who's given credit for transforming hematology from a descriptive art to a dynamic interdisciplinary medical science. So rather than tell you what I thought from reading all of the history, I'm going to you'll have to bear with me, we're going to just quote exactly what Pauling said. This is actually an abbreviated version of what he said 4 years after his paper at the Harvey Society lecture in New York. He said, "One evening, Dr. William B. Castle mentioned to the other members of the committee the disease sickle cell anemia and described the characteristic shape of the red corpuscles and the effect of oxygen in preventing sickling. I pointed out that the hemoglobin molecules in the red cells are involved and that sickling results from an abnormal kind of hemoglobin, which when deoxygenated has the power of combining with itself into long, rigid rods that twist the red cell out of shape." He made this statement, and it's been corroborated, after just hearing this tiny bit of information from William Castle. So how could Pauling jump so quickly to this enormously insightful conclusion with such little information? And this is a true measure of his genius. So I first have to tell you a little bit about Pauling. He was born to a family of very limited financial means in Washington State in 1901. He was born in Portland, Oregon. In 1925, he got a PhD in chemistry and mathematical physics from the California Institute of Technology. And then he won a Guggenheim Fellowship to go to Europe for the period 1926 to 1927, where he studied the new physics of quantum mechanics in Munich with Sommerfeld. And there he met all the giants of physics of the day. There he learned Heisenberg's version of quantum mechanics. He learned Schrödinger's version of quantum mechanics. And in fact, one of the things I learned about Pauling recently, we always associate him with chemistry, but he actually wrote down the first theory of multi-electron atoms beyond the hydrogen atom. Pauling was the first guy who wrote down the quantum mechanical theory, and he did it in both the Heisenberg and the Schrödinger formulation. And most people in those days couldn't even understand the Heisenberg formulation, and Einstein was quoted as saying it was a mystery to him. He then came back and he joined the Caltech faculty in 1931, and in the middle 30s, or actually the early 30s, he began his research on biological molecules, especially normal hemoglobin. In 1939, that's when he published his famous book, Nature of the Chemical Bond, which is one of, or the most influential chemistry books of the 20th century. In 1946, right after the end of the Second World War, he began his anti-war, anti-nuclear weapons activism. 1949 was the paper. In 1951, Pauling discovered the basic structural elements of proteins, these so-called alpha helix and beta sheet, which is the geometry of how the polypeptide folds itself inside of a protein. In 1952, he proposed a wrong structure for DNA. He's famous, actually, for proposing this wrong structure. He proposed a triple helix. We now know it's a double helix. And there was a famous meeting in London in 1952 where Rosalind Franklin presented her DNA diffraction patterns. And anybody who knows anything about Pauling would argue that he would have immediately seen that it had to be a double-stranded molecule and not a triple-stranded molecule. But he was a suspected communist. This was a black period during the 1950s, almost as black as what we're going through now. And he was a suspected communist. And because of that, the US State Department denied him a passport. And he couldn't go to the meeting. In 1954, he won the Nobel Prize in Chemistry for his pioneering work using quantum mechanics to understand and describe the way atoms join to form molecules, what we call chemical bonding. In 1962, he won his Nobel Peace Prize for his opposition to weapons of mass destruction. And during the latter period of his life, he advocated mega doses of vitamins to improve health, especially vitamin C. And of course, this didn't turn out to be correct, and it kind of damaged his reputation in the non-scientific community. This is just a photograph of biographies that I have. I have 8 of them, but I think there are more, just to show what a famous guy he was, that there's already at least 8 biographies written about him. So what I'm trying to explain here is how Pauling was actually prepared for drawing these conclusions when he first heard about sickle cell anemia. It turns out that, as I mentioned just earlier, that he was interested in hemoglobin in the 1930s, and he wrote this paper, which I'm going to describe in a second, in 1935, which is one of the most brilliant scientific papers I ever read. In 1935, when he wrote this paper, these were the two facts he knew about hemoglobin. He knew that it had 4 hemes, which bound the oxygen to the iron in the center of this organic structure. And he knew from the density of the molecule, the density of proteins, and the molecular weight of the protein, that if it were spherical, it had a diameter of about 5 nanometers or 50 angstroms. So these are the two facts. In this paper, which he called The Oxygen Equilibrium of Hemoglobin and Its Structural Interpretation, he was interested in interpreting this binding curve. So this is a measure of how many molecules of oxygen are bound to the hemoglobin as a function of the oxygen pressure. And this so-called sigmoid shape is what people call a cooperative curve. So what that means is once the first oxygen molecule binds to hemoglobin, it's actually easier for the second. And once the first and second have bound, it's easier for the third, and then easier for the fourth. And similarly, in dissociation, once the first molecule of oxygen dissociates, it's easier for the second, and then easier for the third, and finally easier even for the fourth. So what this does, this kind of curve satisfies its physiological function of binding with a very high affinity in the lungs, but then, because of this curvature in this sigmoidal curve, it unloads the oxygen to the tissues. These points are points measured by Gilbert Adair as a graduate student in Cambridge in 1927. So Pauling became curious, and he wanted to develop a theory to explain this curve. So he made one assumption. He assumed that the 4 hemes are equivalent, and with this single assumption, he could explain this curve with just 2 adjustable parameters. So by assuming that the hemes were equivalent, he reckoned that it had to either have a square symmetric array of the 4 hemes or a tetrahedral array. He rejected the tetrahedral array because he insisted that the hemes be on the molecular surface. This was a mistake. He could have gone to an organic chemist at Caltech and found out you can't add oxygen to a heme exposed to water. It will oxidize immediately and will not reversibly bind the oxygen. But he didn't do that. So he rejected this, but he could have moved the hemes inside the molecule, and this is a cartoon of the actual structure of hemoglobin showing that the hemes are, in fact, arranged in a tetrahedral arrangement. So he stuck with his square array, and this is for President Parisi. I hope he can see the equation. So this was his partition function that he wrote down, and from this partition function, a logarithmic derivative with respect to pressure immediately gives you the saturation curve, and that's his saturation curve, which explains the data beautifully. So Caltech was actually the world center for hemoglobin research during the period 1935 to 1955. Since 1955, there have been 2 world centers. One was in Cambridge, essentially due to Max Perutz, who solved the structure of hemoglobin, and also at Sapienza University of Rome, due to this trio of Alessandro Rossi Finelli, Araldo Antonini, and you'll recognize Maurizio Brunori. Do you recognize him? So the Bible for research on hemoglobin and myoglobin for many decades is this famous book by Antonini and Brunori. This is my copy, which is falling apart because I've used it so much. So back to Pauling's discovery. So in 1945, he heard about sickle cell anemia from Castle, and he became, obviously, he wanted to find out what's the difference. He was a crystallographer as an experimentalist, so he wanted to know the difference in structure between this abnormal hemoglobin and the normal hemoglobin. And he hired a new postdoc who just graduated from medical school, Harvey Aitano, who worked very hard on the problem from 1945 to 1949. And the only structural tools they had that were available to them in those days were sedimentation and diffusion, which measure the size and shape of protein molecules, and they could see no difference between normal and sickle hemoglobin. In 1948, Pauling went to Sweden, where he met Arne Tiselius, who won the Nobel Prize in Chemistry, for his development of an electrophoresis apparatus. And this is just a crude diagram. The basic idea is proteins have a different net charge. They have 4 amino acids that bind positively charged ions, protons, and at any concentration of the protons, called pH, there's a different net charge on the protein. So if you put a protein in a gel in a strong electric field, the positively charged proteins will diffuse towards the negative electrode, and the negatively charged proteins will diffuse toward the positive electrode. So Pauling did this experiment, and he obtained 2 major results, which he published in this 1949 legendary paper. The first was that the globin, he determined that it was the globin, the protein part, not the heme part, the protein part of sickle hemoglobin has 2 to 4 more net positive charges. Normal hemoglobin, under the conditions of this experiment, would diffuse towards the positive electrode. Sickle cell anemia hemoglobin was diffusing towards the negative electrode. And from a study of how protons bound as a function of pH, he determined that sickle hemoglobin had 2 to 4 more positive charges than normal hemoglobin. The second major experimental result was when he looked at the hemoglobin from the parents of the patient with sickle cell disease, he discovered that if the parents were healthy, that they had a mixture of the 2 hemoglobins. So this is the electrophoretic pattern showing that it had both normal and sickle hemoglobin. This is called sickle cell trait. And that was very similar to the electrophoretic pattern when he just mixed 50% of normal hemoglobin with 50% of sickle hemoglobin. So Pauling's 4 conclusions from these 2 results, just these 4 pictures. Sickle cell anemia is due to an abnormal molecule. It's a molecular disease. The globin, the protein part, as I said, has 2 to 4 more net positive charges. And this was quite extraordinary, this one. He argued there must be complementary binding sites on the molecular surface that cause the protein to aggregate, to form his rigid rods. And that the structures of deoxyhemoglobin and oxyhemoglobin, they had to be different. Otherwise, the oxygenated red cells would not be normal. They would also be distorted. And finally, and this is one where he was, I think, particularly proud of, he said this was the molecular proof that sickle trait is a heterozygous condition, and therefore the gene is propagated via Mendelian genetics. That means if each parent has 1 dose of the abnormal gene, there's a 25% probability that a progeny will have a double dose and therefore have the disease. So every one of Pauling's 4 conclusions turned out to be correct. In 1956, Vernon Ingram developed a technique which he called peptide mapping. He was working in Cambridge, developing heavy metal derivatives to help solve the crystal structure of hemoglobin and myoglobin with Max Perutz. And a protein, I don't think I've said this yet, but it's a string of amino acids. There are 20 different amino acids connected by what are called peptide bonds. So in this experiment, what he did was he used enzymes to break the hemoglobin molecule up into little pieces, short peptides, and then do something called 2-dimensional mapping, where he used affinity chromatography, which measures the affinity of the individual peptide for the paper that it's diffusing on, and electrophoresis, which separated the peptides according to charge. And what he found was, after then breaking up the abnormal peptide, which is shown here, into even smaller pieces and doing some very sophisticated chemistry, he determined that the mutation was a negatively charged glutamate to a neutral so-called hydrophobic valine. So since there are two beta chains of hemoglobin, two negative charges to two neutral is a net increase of two positive charges. So he was absolutely correct. As I said, the difference in structure, he said, must be on the molecular surface to cause aggregation. And in 1963, Max Perutz solved the structure of hemoglobin, which showed that this site of mutation was indeed on the molecular surface. Now, what you see here is hemoglobin moving back and forth between two different structures which depend on whether oxygen is bound. So the four subunits are actually arranged in a different way in the oxygenated and deoxygenated form, and that's why the oxygenated form, having a different structure, will not fit into the fiber of sickle hemoglobin. Pauling's conclusion that it was a heterozygous condition and that the disease followed Mendelian genetics was confirmed by extensive family studies by an authentic geneticist, Neal, in a paper called The Inheritance of Sickle Cell Anemia. So now we know that the disease results from a single base change in the beta globin gene of DNA, which I will tell you about in a second. So the structure of DNA was solved in 1953 when Francis Crick was a graduate student in physics in Cambridge, and James Watson was a visiting postdoctoral fellow who did his PhD in biology. And this, I think all of you know, but there may be some poets or historians who haven't paid attention to this story. So what they discovered was that the four bases of DNA, so-called adenine, thymine, guanine, and cytosine, pair up along the double strand such that adenine only bonds to thymine and guanine only binds to cytosine. And of course, this is the mechanism for genetic replication. So it's the sequence of these bases that codes for the sequence of amino acids in proteins. And it does so by using a triplet. It's triplets of bases that code for each of the 20 amino acids. So in sickle cell disease, the triplet that codes for the negatively charged glutamate of normal hemoglobin is guanine, adenine, guanine. The single mutation is from adenine to thymine. It becomes guanine, thymine, guanine, and that codes for valine. So changing T back to A would, in fact, cure sickle cell disease. And as I'll say more about in a moment, this is what gene editing is all about. It's just changing this one base from T back to A, and that will cure sickle cell anemia. Now, we've learned a lot about other inherited disorders with a known molecular basis since sickle cell anemia from the Genome Project. This is a slide that was kindly given to me by our director, Francis Collins at the NIH, which shows the growth in the number of inherited disorders that are caused by mutations in the DNA, and now there's something like 6,500 of them. This was the Human Genome Project, which started in 1993, and this is what molecular medicine is about all is today. So really, the modern view of molecular medicine is the result of determining the entire human DNA sequence. When the project started, it was incredibly expensive, and in 2002, it cost $100 million to completely sequence DNA. It now costs $850. And this is because of what's so-called next-generation sequencing, where single molecule techniques have been used, and one can rapidly sequence a person's DNA. And it's had a huge effect. So molecular medicine, based on the sequence of DNA, holds enormous promise for several aspects of modern medicine. First, as I already pointed out, It can cure a disease by gene editing, in this case, using the method now that everyone's excited about. And you'll recall from this beautiful lecture from Emmanuelle Charpentier last year, using this CRISPR-Cas technology. I just mentioned that replacing GTG with GAG in the beta globin gene will cure sickle cell disease. And at the NIH, my colleagues are working on this using CRISPR-Cas9 technology. You can also cure diseases like sickle cell disease by adding a new gene with a virus. This is gene therapy now, where you infect the stem cells. You take out the stem cells of a patient with sickle cell disease, and you infect them with a virus, which is a modified virus of HIV, interestingly, and you load up that virus with genes that are carrying genes for normal hemoglobin. And these take over in the bone marrow. You inject them back. The cells, you actually inject them in the veins. They find their way to the bone marrow. And my colleague John Tisdale has actually cured several patients using this technique already. Then another result of the Genome Project is to find genes as targets for drug therapy. So, for example, in the case of sickle cell disease, there's a gene, they call it BCL11A, doesn't matter what the name is, that actually restarts fetal hemoglobin production. Fetal hemoglobin is the hemoglobin you're born with, and it inhibits sickling. So by suppressing this gene that's suppressing the fetal hemoglobin, it's possible to obtain a cure for sickle cell disease. One of the most interesting is that from so-called genome-wide association studies, from correlations between DNA sequences in disease, we now have something called personalized medicine. So this will prevent disease by predicting susceptibility and guide the treatment rather than this one-size-fits-all approach of Western medicine up to now. So the one you all know about is mutations in a gene called BRCA1, which is associated with an increased incidence of breast cancer. So women who have this mutation are advised to get surgery because they're almost certainly going to get breast cancer at some point. But I think one of the most interesting is a protein that Professor Brunori knows a lot about, is the differences in the activity of a drug metabolizing enzyme called cytochrome P450. Cytochrome P450 is an enzyme in the liver that modifies drugs. And it can destroy the drug, or it can actually modify a molecule into a drug. Almost everyone has a different sequence for cytochrome P450. And that means that if we know which of the forms of cytochrome P450, it's actually going to guide physicians on the dosages of medicine that we take. So I think that's one of the really interesting aspects of this molecular medicine approach. So current research on treating sickle cell disease by gene therapy are important to know because the current gene therapies require very advanced medical facilities. They cost over $100,000, so they're not going to be available for decades, I would argue, for over 95% of the patients in the world because they live in third world countries. So so my introduction to this other approach to treating sickle cell disease is to answer these two major questions unanswered by Pauling. What's the mechanism of Pauling's long, rigid rod formation, we now call the rod a sickle fiber, and how can the disease be treated other by gene therapeutic approaches? So elucidating a mechanism is based on studies of kinetics, that is, watching sickle fibers form as a function of time. The fibers actually, I'll just show you a picture of the fiber. This was solved by a biochemist, Stuart Edelstein, in the 1970s, and this is the structure of Pauling's long, rigid rod at molecular evolution. and molecular resolution. When we studied the formation of these fibers, we found these incredibly unusual kinetics, that there is a lag phase or a delay time before the appearance of any fibers after the oxygen was dissociated from the molecule. And in addition to this lag phase, or this sharp delay time, we found an enormous concentration dependence, actually to the 30th power. And what that means is that if one decreases the concentration by 8%, this delay time increases by a factor of 10. In a normal chemical reaction, a bimolecular reaction, if you decrease them by 8%, then this curve will shift by 8%. But you see an order of magnitude increase. When we tried to, we knew it was some kind of nucleation mechanism, but the only one that was available at the time that we knew about, or anybody knew about, was due to the famous Japanese biophysicist Fumio Osawa, it could not explain our data because it couldn't explain this sharp delay. So we had to invent a new mechanism, which we called a double nucleation model, and this was done when Frank Ferrone was a postdoc in the lab, and Jim Hofrichter had just become a new principal investigator in the lab. And the basic idea is that the first molecule, the first fiber that forms in a given volume of the solution forms by the Osawa mechanism, but the additional fibers which eventually dominate that are responsible for this delay period, which we call heterogeneous nucleation, is when this critical nucleus forms on the surface of pre-existing fibers. So secondary nucleation on the surface of pre-existing fibers was the key to understanding the mechanism of sickle fiber formation. And interestingly, this is exactly the same mechanism that's being now used in Cambridge to explain the aggregation of the Alzheimer's peptide. So it adds some historical significance to this work. But the more interesting part, at least for me, was the relevance of these highly unusual kinetics to the disease pathogenesis. And what we recognized early on is that this delay time actually allows the majority of cells to escape these narrow vessels before the fibers form, and this is what makes the disease survivable. And a single concept synthesized an enormous amount of clinical information, the concept being that clinical severity is determined by the length of this delay before the fibers form relative to the capillary transit time. So it's this delay relative to the time that it takes to pass through these narrow vessels that determines the disease severity. So if you get a fever, there's a very high so-called activation energy, which shortens the delay time and makes it more probable that the cell will sickle inside of the capillary. If, however, the delay time, the delay time for fiber formation remains the same, but the transit time through the capillary is shortened, that also increases the probability that sickling will occur and possibly cause a logjam effect. So what do these kinetics tell us about how to treat the disease? Because this was the major question that Pauling left unanswered. In fact, when he lectured at the University of Pennsylvania School of Medicine when I was a student, his only answer to the question was to advise patients not to marry if they were both heterozygotes because there was a 25% probability they'd have a child with sickle cell disease. It was not a very politically correct thing to say, but Pauling said it anyhow. So what the kinetics tell us is that it's not necessary to completely prevent sickling, but just to increase the delay time before fibers form to allow more cells to escape the vessels of the tissues. So what we want to do with a drug is we want to increase the delay time so we go from this scenario to this scenario. This is a summary now, and I've already said part of this is the current state of therapy for sickle cell disease. It can be cured at advanced medical facilities by stem cell transplantation with a matched sibling donor. The most successful has been John Tisdale, my hematology colleague at the NIH. The promising new curative methods, such as gene editing, even if eventually successful, will only be available for a small fraction of patients for many years. There's only one drug for sickle cell disease called hydroxyurea that is an anti-sickling drug, and what it does is it induces fetal hemoglobin synthesis. So by replacing the sickle hemoglobin with this non-polymerizing fetal hemoglobin, this increases the delay time enormously, and so the effect of hydroxyurea is actually its effect on the kinetics, but it's only partially successful because it doesn't induce fetal hemoglobin in all the cells. It only induces them in maybe 60 or 70% of the cells, which leaves a large fraction of the cells that are pure hemoglobin S that will sickle very rapidly. So what's urgently needed is an inexpensive drug that increases the delay time in all cells and can be taken orally in order to make it available worldwide. So my current research now, after all these years of basic research, is to find such a drug by a high throughput screening of drug libraries using a kinetic assay. And this is just about the end of what I'm going to tell you, but I guess you always run it. Oh, isn't that interesting? Microsoft does it again. Nope, will not advance. That is amazing, isn't it? No, it's frozen. It's frozen. Unbelievable. All right, let me try Control-Alt-Delete. No, it will not. Nope. Unbelievable. Okay.
Sta lavorando.
Oh, just.
Non vede più il computer.
No.
No, infatti, perché sta lavorando.
Okay, so.
Non può, no. Qualcosa succede, ma è il computer che si è un po' impallato qua, vedi? Infatti.
No.
No. Working.
Yeah. Okay. I thought I was going to be killed by Microsoft. I was only wounded. Okay. Yeah.
Guardi.
I need this. Thank you.
No.
Oh,
Please.
what is this? Where's the pointer? I can't see it. Oh, here it is.
Switch music.
Yeah.
Chiudi. Close.
I'll get here in a second. Let me just do the slideshow.
From current slide.
Oh, no. Okay, sorry, I'll just have to go through that. Okay, don't touch anything. It's going to work. It's going to work. It's going to work. It's working. It's working. It's working.
Vado. Un po'. Non vede. Vede, non lo chiuda. Oh.
Nope, it's stuck. It's stuck again.
Stop again.
Unbelievable.
Si blocca il suo computer. I don't know.
Okay, I'll give it one more try, then I'll do it without slides. Okay. That's a real bummer. What's that? You know what I'm going to do? I'll do it without the slideshow. This is good enough. This is what I'll do. No slideshow. Okay, yeah. This will do it. This is not ideal, but this works, I think. Okay, sorry. This one, everything has happened to me in lectures, but this one has never happened. Okay, so what we're doing is high throughput screening to find a drug. And the most precious library for drug screening that everyone should know about, because it's a very interesting library, are 12,657 compounds that have been tested by humans. And this was financed in part by the Bill and Melinda Gates Foundation. So any drug that shows therapeutically significant anti-sickling activity in our screen at concentrations known to be non-toxic in humans can be tested in clinical trials almost immediately at the NIH on sickle cell patients. We have all of the infrastructure at the NIH to do this. And of course, we'd have to get approval from the FDA and the IRB, but that could go very rapidly because the drugs have already been given to humans. So our assay is a kinetic assay where we take videos of cells after we remove or as the oxygen is being removed. And this video, I think I'll come back to the video at the end just to make sure that I can tell you the final conclusion. I'll show you the assay in a second. But basically, we've just screened all 12,657 compounds between 1 nanomolar and 10 micromolar. And we have what they call in the pharmaceutical industry 167 hits. That's a lot of hits. But moreover, depending on the concentration and knowing the statistics of what concentrations of drugs are found in the human serum, we think we now have approximately 30 new candidates for drugs for sickle cell anemia with this approach. And I'd like to end then by just showing you this video, because I think it's a beautiful demonstration of our assay. Oh, no, just a second, come on. This is unbelievable. Okay, let me go down.
Can explain it to the audience?
Yeah, I will. I'm going to explain it. I am going to explain it. And, okay. Okay. So what you're seeing here, these are red cells before we remove the oxygen. And what you're going to see is that the contrast increases, which is an indication that the cells have lost their oxygen. And you'll see the cells change shape. So watch these 4 cells up here. You see that one just sickled, that one just sickled, that one just sickled. So what we do then is we basically, our candidate drugs are those that increase this delay time before sickling. And that's the way the assay works. And as I said, we have 30 candidates, so wish me luck. My NIH co-workers on the basic science studies, the key guy was Jim Hofrichter. The early basic science work was done by Phil Ross, Helen Sunshine, Frank Ferrone, who's a professor of physics at Drexel University. He's gone on to refine the kinetic model and done a lot of brilliant work on sickle cell disease. Some of the very important early work, one was experiments with Massimo Coletta, who is a student of Maurizio Brunori, and Andrea Mazzarelli, who is a student of my dear friend John Luigi Rossi in Parma. So, and then today, the key guy now in our current assay is Eric Henry, Jim Huffrichter is still involved, a series of very dedicated coworkers, and getting samples from patients is really important. So we've had fabulous cooperation by people at the NIH who've supplied us with samples who've allowed us to do this drug screen. So sorry for the delay. It's not my fault. And thank you for your attention.